So the work I'm going to present you today is part of my PhD. So starting by the observation that animals produce...
You can remove your mask here.
That's it. Wide variety of signals, both in nature and a number of different signals. And this has been conceptually and empirically also attributed to different factors, including, as Alban mentioned, the sociality. And this was formulated under the social complexity hypothesis for communicative complexity that suggests, again, I think you did a nice introduction to my talk, that animals facing a higher diversity of potential partners and different social interaction will have the need to produce and recognize a wider range of signals and also to do so in more flexible ways. And this hypothesis has been tested with correlative tests mainly in many different taxa, finding a lot of supportive results, but also, and usually more recently, also non-supportive results or non-significant differences. And this is partly because both social and communicative complexity have been assessed using a wide range of different proxies and also at different levels, so from species to the interaction. And the tests, on the contrary, are made more specifically using usually one specific proxy for social complexity and linking it to one specific proxy for communicative complexity. But the conclusions are usually made at the broader level, which makes it difficult. And the actual debate on how to best quantify social complexity argue for a more comprehensive approach of it, including all the dimensions of social systems, to not dismiss some of these aspects. And we here argue that the same approach is also necessary for the communicative complexity. So communicative complexity has been assessed via two main dimension. The first is the number of signaling units, so for instance the repertoire size or the rate of expression, and the other dimension is the amount of uncertainty, so for instance the number of signal combination or the individuality in a signal. And one study saw that, and one of the really few studies that consider both a comprehensive approach of sociality and communicative complexity is the one of Boucher and colleagues. But when talking about communicative complexity, they only consider the vocal modality. And we here argue for also the importance of integrating the multimodal signal or the multimodal aspect of the repertoire. So not only the vocal aspect. And this is because maybe this hypothesis is true. So for each different dimension of communicative complexity and each modality, but it could also be that a possible compensation strategy or alternative hypothesis exists. For instance, only an increase in the number of signaling units, but not in the amount of uncertainty. And this can be true only in one modality or in all modalities. So to approach this question, we try to adopt this comprehensive approach in the lemur. So lemur also because they rely a lot on three different modalities: the olfactory, visual, and vocal modality. And they also are closely related species that have diverse social systems. And in particular, we choose to compare two closely related species, the lemur rufifrons, that are multi-male, multi-female, live in multi-male, multi-female groups and are egalitarian, with a pair living species exhibiting female dominance. And so the prediction is because the lemur rufifrons are considered to be living in more socially complex environments, they are supposed to also, or we expect to find also more complex communicative complexity. So to do so, we went in Madagascar in two different field sites, and we collected data on three groups of each species. So we did some focal recording and also some focal behavioral observation for the visual and olfactory signals. And so what we obtained is that we could identify different numbers of signals across modalities in these two species with really many more signals in the lemur reference, so the more socially complex, and this across the 3 different modalities we studied. We also found that the signaling rate were higher in these more socially complex species in both olfactory and vocal modalities, and that the number of signal combination was also more important in this species, and this across the different modalities. But we wanted to go a bit further in this, and we wanted to really have an approach at the system level. So we decided to base our approach on a study on birds, Wilkins and colleagues, and to have a network approach. So here each dot is a different signals, the color and the modalities, and the edge of the network represent the predictability of the behavior between these 2 signals. So for instance here, an individual that would exhibit a lot this signal would also exhibit a lot these 3 different signals. So building this network for our 2 species of interest, we obtain these networks, and based on this network we assess different parameters, first being the average correlation, so how strong are the edges that we observe, which means that the stronger are the edges, the more predictable is the system. And we found a positive significant difference between the 2 species as predicted. We also look at the edge density, so how much we can predict, how many edges we found, and here we had no significant difference between the 2 species. And finally, we also assess a third parameter, that is the assortativity coefficient, that is how much these signals are connected within or between modalities, or what would be the more uncertain is randomly across and within modalities. And here again, we couldn't find any significant difference between the 2 species. So again, we have the only significant difference we observed was in the direction of the prediction. Now looking at the non-homologous signals, so the ones that are not present in the 2 species, we could only find these signals in the lemur reference. So these non-homologous signals are here in yellow, so could have been retained from the ancestral state and be non-homologous signal, or what is also called derived signal that were not present in the ancestral state and evolved later. So here we have these 2 scenarios. Interestingly, in the lemur, the ancestral state is a group-living species exhibiting female dominance, meaning that this non-homologous signal here may have been lost in the lemur mongoose when pair-living style evolved, so with a reduction of the pressure associated with this group-living environment. Or, on the contrary, this signal could have evolved in the lemur reference with the apparition of a more egalitarian society. Now, to distinguish these 2 scenarios, we would need to look at the more specific function of each of these non-homologous signals. Or also, we could try to study more species, and especially the ones that have the ancestral state of group living with female dominance. So here I argue for the adoption of a more comprehensive approach to communicative complexity based on the 2 dimensions of communicative complexity and including the multimodal aspect of communication. And also to develop measure at the system level. So for example, here to take those 3 measures of the network, we could build a complexity space, 3D complexity space, in which whole species could be replaced. And we could imagine to do so because it's based on the rate of expression of the signals, so something quite easy to collect. We could think to include or develop this method to include other taxa also, which I'm trying to do now for my postdoc. And this would be interesting to enrich the discussion about also the difference between inter-individual variation in terms of sociality, like existing primates, or what we find more in insect society with more different social roles. So this could bring some insight into this discussion. And with this, I want to thank the local team and all the person that made these studies possible, and you for your attention. If you have any questions.